Hello friends, welcome to my channel Star Biology Class. Today we will discuss about the principle of electron microscopy and its applications. Before that, will you please consider subscribing my YouTube channel. Your subscription and viewership is often a lot to me. An electron microscope is a high-resolution imaging instrument that uses a beam of fast-moving electrons rather than light to illuminate the specimen. Unlike light microscope, it uses a series of electromagnetic lenses in order to focus the beam of electrons onto the specimen and to magnify the image. Electron microscope can have a maximum magnification of up to 50 million times. As the wavelength of an electron is more than 10 to power 5 times smaller than that of visible light, electron microscope have a much higher resolution, about 0.1 nanometer. On the other hand, the resolution of a light microscope is approximately 200 nanometer. Electron microscopes have several applications. Number one, biological research and medicine. It includes microbiology and cell biology, diagnostic pathology, and vaccine development. Electron microscopes are used for imaging viruses, different types of bacteria and other cells, and intracellular organelles. It is also used to understand the mechanism of various diseases at the cellular level and to test and examine new medicines and vaccines. Number two, material science and engineering. Electron microscopes are widely employed to characterize nanoparticles and nanomaterials at the atomic or molecular level. In addition, it is also used to examine metals, alloys and crystals to identify their defects. Number three, quality control and forensic science. It includes quality control, forensics and geology. Electron microscopes are widely used in quality control, in identifying evidences such as gunshot residues, fibers and other biological particles at the crime scene, and also to analyze the chemical composition of soil and rock samples. Components of an electron microscope. Number one, an electron gun. It is a heated tungsten filament that generates electrons. Number two, an anode. What is anode? It is a positively charged metal plate or an electrode which attract these electrons, accelerate them and shape them into a fine electron beam, which is then directed downward the column of the microscope. Number three, electromagnetic lenses. Number three, electromagnetic lenses. As already said, instead of glass lenses, an electron microscope uses electromagnetic lenses to focus the beam of electrons and to magnify the image. The electromagnetic lens utilizes the magnetic field generated by an electron carrying copper coil. In electron microscope, three types of electromagnetic lenses are used. A. Condenser lens. Condenser lens. The electromagnetic condenser is used to focus the beam of electrons onto the specimen. Next one is the objective lens. The function of the objective is to collect the electrons coming out of the specimen and forms the intermediate magnified image. Third one is projection lens, which is similar to eyepiece. This is the projection lens, which further magnifies the image formed by the objective lens and produce the final magnified image. Another component is specimen holder. The specimen holder is an extremely thin film of carbon or colloidal supported by metal grid. Last component is image viewing and detection system. The final magnified image is projected onto a fluorescent screen. Below the screen, there is a camera for recording the image. The whole microscope is mounted vertically in a vacuum column so as to prevent the collision of electrons with air molecules. So in the electron microscope, three different lenses such as condenser, objective and projector work together to produce a highly magnified image which is more sharper, clearer and detailed. There are two types of electron microscope. Number one, transmission electron microscope, and number two, scanning electron microscope. Transmission electron microscope, TEM. The fundamental principle of a transmission electron microscope is very much similar to that of a bright field light microscope. In both, radiation is transmitted through the specimen to form the two-dimensional image. In both cases, denser areas of the specimen which absorb or scatter more radiation create contrast. On the other hand, thinner areas of the specimen through which more radiation passes unhindered appear bright in the image. Both uses objective and eyepiece to produce the magnified image. In both of them, condenser is used to focus the radiation onto the specimen. However, there are certain differences. In transmission electron microscope, a beam of fast-moving electrons is used to illuminate the specimen. On the other hand, in light microscope, visible light is used to illuminate the sample. Another difference is that in case of electron microscope, electromagnetic lenses are used. By contrast, in light microscope, glass lenses are used to focus the radiation and to magnify the image. This figure illustrates the principle of a transmission electron microscope. First component is an electron gun, which is a heated tungsten filament that generates electrons. The electrons which are generated by the gun are collected, accelerated and shaped into a fine electron beam by an anode which is placed between the electron gun and the condenser. The anode is a positively charged metal plate or an electrode. The function of the condenser lens is to focus the fine beam of electrons onto the specimen. When the electrons strike the specimen, denser areas scatter more electrons. On the other hand, more electrons passes unhindered through the thinner areas. The transmitted electrons which passes through the sample is then collected by the electromagnetic objective to form the intermediate magnified image. The intermediate image which is formed by the objective is further magnified by the projection lens which forms the final magnified image, which is then focused on a fluorescent screen. In addition to this, the image can also be recorded by a suitable camera. The function of the transmission electron microscope is to study the internal structure of virus, different types of bacteria, plant and animal cells, and also tissue sections. Number 2, scanning electron microscope SEM, which is used to study the surface topography of different types of specimen. This figure illustrates different components of a scanning electron microscope. First one is the electron gun, which generates electrons. The electrons are collected, accelerated, and shaped into a fine electron beam, which are then focused onto the sample by the condenser lenses. These are two condenser lenses whose function is to focus the electron beam onto the specimen. Another component is a scanning coil, which systematically deflects electrons onto the surface of the specimen in a synchronized rectangular pattern known as raster scan. This coil is controlled by a scan generator which continuously monitors the position of the electron beam along x and y axis. This positional data is directly fed into the computer of the system. Other components of the scanning electron microscope are the objective lens, electron detector, sample holder, amplifier, and computer. When the electron beam strikes the specimen, two types of electrons are emitted from its surface. One type of electron is known as secondary electron, which is of low energy. These electrons generate weak electrical signal, which is recorded by the detector. This weak electrical signal is then amplified into a strong readable voltage. The function of the scan generator is to synchronize the positional data with the electrical signal that is amplified to produce the detailed pixel by pixel image on the system. Secondary electrons are of low energy. Because of this, these electrons deep within the specimen fail to escape. Only the secondary electrons that are emitted from the surface reach the detector, which is then converted into weak electrical signal. The signal is then amplified into a strong readable voltage, which is used along with the positional data to form a detailed three-dimensional image of the surface topography. Another type of electron emitted from the surface of the specimen is backscattered electron. These are of high energy and are recorded by the detector, which is used to distinguish different regions within the sample based on their elemental composition and atomic number. So scanning electron microscopy is widely used to study the surface details of various specimens in a precise manner. So friends, this is all about electron microscopy, principle and applications. For more updates, subscribe my YouTube channel, Sam Biology Class.